talking about we can do as extra credit?
Yeah, so there, it says, if you click on syllabus on Canvas, there's a list of all the stuff that's due. And then it'll say extra
What's
credit.
due for what's due tomorrow? That doesn't count for us?
Yeah, so you've already done it. So yeah, so it won't, like, you guys will get points for going to that lab, and they're gonna get points for doing it too. So you guys can't double do that because they can't either. So they'll get graded on theirs too. But the other thing you can't. Okay. So remind me where we stopped. Because I'm getting old. I can't remember. Was it here? Was it here? I think it was here, right? We're talking about the blending theory of inheritance.
Yeah.
Nobody remembers.
We already talked about the flowers.
Yeah, we talked about that, and we
We're talking
talked
about
about,
these.
So we're past this?
we were just starting that, yeah.
We're here?
I think on the.
We're here? I thought we were here.
I think so.
Yeah.
Oh, heck, we'll just start here.
Yeah, because I think you finished talking about the flowers and we were asking something weird about the flowers.
All right, so,
Let's see.
okay.
Thank you.
Everything's blinking, so we're recording. So let's do a little review. And like I said, I'm gonna spend the first like 15 minutes or whatever talking about this, and then we'll do the review. Okay, so we have we talked about Mendel's crosses between the purple and white flowers. You guys remember that?
Yeah.
So we have a purple one, and we cross that with a white one, and that we called that the parental generation. And then they had babies, and that Mendel planted those. And you remember what color they all were, right? And then he let these self-fertilize, and they had offspring, and that's the F2 generation. And so, do you remember what these were?
3 purple.
Yeah, 3 purple to 1 white. And remember, this means 3/4 and 1/4, but we don't really write the fractions. We just write it as a ratio like that. So it's 3 to 1. Okay, so now you guys already know from the meiosis chapter and the mitosis chapter that we and plants are, they're 2N, so what's the term for that? What do we call an organism that's 2N? Like us.
It's
Diploid.
Yeah, diploid, right? Or diploid. So these plants are diploid too, which means that they have 2 chromosomes, one they got from their mom, one they got from their dad, and on there are alleles, and those alleles code for a gene. In this case, it's the flower color. And so this one we would say is true breeding. That means that it has both alleles for purple. So this chromosome we got from its mom makes a protein that reflects light in the purple wavelength. This one does the same thing. It makes a protein that reflects light in the purple wavelength. So we would say that that allele is big P and this one's big P. The reason that we label it that way is because we use the first letter that's capitalized in the dominant trait. So it's purple, so that would be capital P. And then these are little p's. So we still use the dominant first letter, but it's lowercase. And here we would have 2 alleles, but they're broken. They don't make anything. Remember, the reason they're white is because the default color is white, just like the walls and, if you move into a new house. So that could happen anywhere. It could happen here, so that the DNA is never made into RNA, And what would happen then? You'd never get a protein, right? Or it could be made into mRNA and there could be a mutation so that doesn't fold in the right shape and then it doesn't reflect light in the purple wavelength. So all kinds of things could happen where this wouldn't function, but regardless, it doesn't work. So we symbolize these broken alleles as little p, and the what we see is white. So remember the law of segregation means that you're going to make gametes and that these have to segregate from one another because we're going to make the gametes as end. And what is that term for the end? Haploid. Excellent. So what gametes, let's say that this is the male and this is the female. So what gametes is this male going to make? What can it make?
Big P.
Big P or big P. That's it. And what gametes could this female make? So it can make little p or little p. So this sperm fertilizes this egg. What do the chromosomes look like in this offspring? Big P, little p, right? And we cross that with itself, so we know that this cross is big P, little p times big P, little p. So what gametes can this one make? We'll say this side is male. Big P, put a tail on it for a sperm, and little p. This one makes big P or little p. And so this sperm fertilizes that egg. What do we get? This sperm fertilizes that egg. Big P, little p, correct. This sperm fertilizes that egg. Right, it's little p, big P, but we'll just flip it. Or that sperm fertilizes that egg. So this one's genotype, what its genes are, is big P, but what is its phenotype is what it looks like. So what color is that? Good. So its genotype is big P. Its phenotype is purple. This one's genotype is big P, little p. What's its phenotype?
Purple.
This one,
Purple.
and then this one.
White.
So we get that 1, 2, 3 to 1 ratio of purple to white. Mendel figured all of this out without knowing anything about chromosomes or segregation or gametes or any of this stuff. So it's actually pretty amazing that he did this all based on that 3 to 1 ratio that we see. So this screen just shows you some terms that you need to know. So these, because they have the same alleles, we say that those are homo, right? That's the term for same. These are the same too. So that's homo. And then when we're talking about genetics, we're talking about what is it like in the fertilized egg, because everything that derives from that egg is the same. How does the egg divide? Mitosis, so it's genetically identical. So all we have to do is distinguish what the zygote looks like. So we say that this is homozygous, because that's what it looks like in the zygote. This is also homozygous. So we can't distinguish this one from that one by using that term. So we have to say that this is homozygous dominant, homozygous recessive. And then this is heterozygous. And that's the term. And then what you see is the phenotype. So this is the phenotype, purple and white. And then what you can't see, right? If I asked you to walk outside and pick a purple pea plant, would you know if it's this or that? You couldn't, unless you had some special vision where you could look at its genes and tell, yeah, that's a heterozygote. Nobody can do that. So you can see its phenotype, but you can't see its genotype. You would have to do some sort of testing to see what its genotype was. So any questions about these terms?
So on the third one down, like on the second one, how it's gonna be the hetero, and then down,
This one?
and then go down one more. If it were to end up being big P again, then it would go back to being considered homo.
Wait. This offspring that's big P,
Yeah,
yeah, that one's like the parent.
but it would go back to being considered homo because it's big P.
Yep.
Yeah, so that's
So
homozygous,
it
yes.
doesn't matter that the parent was hetero?
It's what you are, right? Whatever you're talking about is what the term refers to. Does
Okay.
that make sense? Okay. So I did all this, you know, this sperm fertilizes this egg and so on and so forth. But this guy named Punnett, you know, the joke is that his real name was Square because it's called the Punnett Squares. But basically, this is just a way to line up the gametes. So on one side you have the males, the other side you have the females, and you just make a box for the number of gametes. So this one made this. Remember we said we were going to consider this one to be male and this one to be female. So what gametes can the male make?
Big P, a little P.
It's either big P, right? Because these have to sort independently. You can't have 2 in 1 sperm. That would make it too many chromosomes. So this represents the big P sperm. This represents the little p. This one, the eggs, right? Because that's what females make. Big P or little p. So this sperm fertilizes that egg. What's our zygote?
Big P, big P.
This sperm fertilizes that egg. What's our zygote? This sperm fertilizes that egg.
Big P.
I mean, it's little p, big P, but we just flip it around. And then this one fertilizes that egg. So that's the same thing that I just did. But this is much easier because sometimes you have to deal with more than one trait. So if you have 2 traits, then you can make 4 different gametes. Because the rule is it's 2 to the n power, where that is the number of traits. So how many gametes could this make? How many traits do we have? It's 1, right? It's flower color. So 2 to the first is 2 times 1 is 2. If we had 2 things, let's say we're talking about flower color and seed shape, then that would be 2 to the second, which would be 4 gametes, right? And if we're talking about flower color, seed shape, and plant height, which we're doing in the lab, then that would be 2 to the third, which would be 8 gametes, right? So if you had 3 traits, you would need 8 things here and 8 things over here. So how many squares would you have? 64, right? So this thing can get really complicated really fast. Remember, we have 20,000 different genes. So how many would you need for that? 2 to the 20,000th power would be the number of gametes you can make, right? All right, so the Punnett square is just an easy way to do this. Remember that, so one of the most important things that you can remember in genetics are these ratios, because they're going to help you do shortcuts that you otherwise would have to construct a Punnett square for. So what's the ratio? We call this right here a heterozygote. Another term for a heterozygote is a hybrid. So that cross is called a monohybrid cross because it's between 2 heterozygotes for 1 trait. If it were 2 traits, we would call it dihybrid. If there were 3 traits, what would it be? Trihybrid. 4 traits, what's the word for four? Yeah, tetra, hybrid, and so on. So the ratios for a monohybrid cross are, for the phenotype are what? 3 to 1, right? 3 dominant to 1 recessive. Right? 3/4 to 1/4, or 75% to 25%. So this is the phenotype. What's the genotypic ratio? Is it 3 to 1? Yeah, it's, well, I erased it, but it's big P to big P little p to big P little p to little p, so it's 1 to 2 to 1. 1 to 2 to 1. Do you get it? Homozygous dominant, heterozygote, homozygous recessive. So that's what this shows. Genotypic ratio is 1 to 2 to 1. Phenotypic ratio is 3 to 1 for a monohybrid cross. All right. So let's say that you didn't know what the organism was. So remember my example, I sent you outside, I told you to pick a purple pea plant flower. You bring it to me, and I ask you, what's its genotype? Can you tell me? Is there a way you can figure it out? How?
Look under the microscope.
Well, you can't see genes under a microscope.
How do they see that?
You cross-pollinated again?
You could cross-pollinate. What would you cross-pollinate it to? You would want to cross-pollinate it to something that you know absolutely for sure when you looked at it what its genotype was. So what's the only one you know for sure? Yeah. So we call that a test cross. I mean, they did this back in the day, right? Let's say that you wanted to spend $4 million on what's a thoroughbred? It's a horse. But what does that mean, thoroughbred?
Hadn't been crossed with any other type.
Right, it's a true breeding, purebred line. But you could get a heterozygote, right? You wouldn't see the difference because if that was a dominant trait, then some body could pass off their heterozygote on you. Would it be a thoroughbred then? Would it be worth $4 million? No. So how would you know? You'd have to do a test cross, right? Just like we would do with the flowers. So this is getting really complicated. I'm gonna erase this. But test cross is really simple. It can be this, right? The purple flower. Let's go back to the flowers because horse genetics is really complicated. So big P, or what other genotype could it be for the purple flower? Big P, little p. And we want to cross that to what we know, which is, all right, so let's say it's this one and we do the cross. What would the offspring look like?
Big P.
Big P from this one, and so they would all be purple. So you know that it's big P if you do a test cross. That means crossing it to something you know, and they're all exhibit the dominant trait. Now, if it was this, and you crossed it, what would you get?
Big P, little p. And 2 little.
What ratio? So we could use the Punnett square, right? We just use big P on one and little p on the other. And so we get big P, little p, little p, little p, big P, little p, little p, little p. So what's the ratio? So just to remind you guys, to figure out these ratios, all we're doing is multiplying the probabilities, right? So just like this, what's the probability that that gamete would be made? 1 out of 2, right? It's the same as flipping a coin. There's 2 outcomes. So what's the possibility of this outcome? 1/2, right? Just like heads. This one is 1/2. So we can write that down here. And what's the probability that this one was be made? 1/2. And this one, 1/2. I'm sorry, 1/2. And so the rule of multiplication says that the probability that this gamete would be made is 1/2 times 1/2, which is 1/4, right? So each of these is 1/4. And then the rule of addition says that I can add these together because they're the same. So that gives me 2/4 would be big P, little p, right? And I can add these together because they're the same. So 2/4 would be little p, and then that reduces to 1/2, 1/2, and remember, ratios are simply the fraction with the denominator taken off, so that's a 1-to-1 ratio. Make sense? Okay. All right, so dihybrid cross, a little more complicated. So let's try this one. Mendel did a bunch of crosses. One of the ones that's pretty routine is he did seed color and seed shape. So on the screen, it shows you that he crossed big Y, and big R, by little y, little y, little r, little r. So what's dominant here?
Big Y.
Well, what's the phenotype? What does that stand for?
Yellow.
Yellow, and that's
Brown.
round. So this is green, and that's wrinkled. You get it? We got 2 traits. That's a dihybrid cross. This isn't the dihybrid, right? This is the parents. So what gametes can these make, these parents?
Big Y, big R.
So remember, it would be like you passing on hair color and eye color. You can't just pass on hair color, so you can't make a gamete that's YY, because then you wouldn't be giving your kids any eye color. Does that make sense? So you had to have one of each. So it can only give one of these, which is the only possible outcome is big Y, right? And it can only give one of these, so the only possible outcome is big R. And then this one is going to make a gamete that's little y, little r. Let's say this is the egg and that's the sperm. They come together and we get big Y, big R, little r, right? And then we self-fertilize these. So what do we call that? What do we call it if it's a heterozygote? What's another term for that? Hybrid. So that's hybrid for 2 traits, so that's a dihybrid. Everybody get it? So this is the dihybrid cross right here. Now the question is, what gametes can that make? Remember the rule, it's 2 because there's 2 chromosomes, and then how many variables? 2, so how many gametes should we come up with? 4. So if we didn't come up with 4, we did something wrong. So what is that? What are the gametes? Big Y, big R. Big Y, little r. Little y, big R. Little y, little r. And then for the mom, same, right? And we can do our Punnett square, which I'm not going to do because it will take forever. But it's done for you out of the book. And so you can see the ratio, and you guys can test this if you want, but the ratio, the phenotypic ratio is 9 to 3 to 3 to 1, where the 9 is both dominant traits. The 3 is 1 recessive and 1 dominant. This other 3 is 1 dominant, 1 recessive, and then this last one is both recessive. So in this case, what are the 9? What's their phenotype?
Big Y, big R.
Nope, phenotype, remember? So it's what you see.
So it's yellow round.
Yellow round, that's right. So these are yellow round. And then this one is, you could make it green and round, that's right. And then this one would be and that one would be that's right. So what about the genotypes? What if I wanted to find out what the probability was that you would get this offspring? How could you do that? Well, in this cross. And the answer is you can do that because of the law of segregation. That means that we can treat this set of gametes different than this set of gametes. So what's the probability if you crossed, if it was a monohybrid cross and you cross this and that, what was the probability you get that?
1 out of 4?
Yeah, you're right, remember, because our genotypic ratio on a monohybrid cross is what?
3 to 1.
No, genotypic ratio is 1 to 2 to 1. 1 both dominant, so that would be 1/4 that you would produce that. And from this one, what's the probability you'd produce that?
1 half.
Yeah, because that's the 2, right? 1 to 2 to 1. So that would be, so 1/4 times 1/2 is 1/8. And you could check, right? In fact, if you look at this, you'll see there's 1/16. 1 out of 16 is this one, plus 1 out of 16 is this one. So add them together, the law of addition, rule of addition, and what do we get? 2/16, which is 1/8. So that's how you do that. So let's try another one. What about if I did this? Cross to what's the probability I'd get this offspring? So let's do it out. What's the probability that we would get big A from that cross? Well, if you're not sure, you can do the Punnett square, right? Big A, big A, little a, that's the cross, so what's the answer? 1/2. So this is 1/2. And then what would the probability be that we would get this one from this cross? Correct? And this one from this cross? Is it 1/2? Big C, little c, little c. What's the answer? 100%, right? So then that would be 1. And then what about this one? It's a half. And this one? It's also a half. So then half times a half is a 4 times 1 is a 4 times a half is times a half is. So the answer is 1/16 chance that you would get that from that cross. All right, so important things that you guys should remember is monohybrid cross, what's the phenotypic ratio? 3 to 1. What's the genotypic ratio? 1 to 2 to 1. And a dihybrid cross, what's the phenotypic ratio? 9 to 3 to 3 to 1. And then you don't need to know the ratios, but you should be able to calculate this based on the multiplication rules that I showed you and the law of segregation. All right. So just to remind you, law of independent assortment allows us to be able to do these individually and then multiply them together later. And it's all based on probability and statistics. So if I flip the coin, what's the probability it'll come up heads? 1/2. If I'm going to make this gamete, what's the probability that I would make this gamete from this individual?
1/2.
How many are there? So it'd be 1 out of 4, right? So 1/4 times 1/4, what's the probability that I'd make this individual? 1/16, right? Everybody get that? And then we could add them together if they're the same, and that's the rule of addition. So I could add, well, this is multiplication, but I could add these together. If this was big P, little p, then I could add them together, and that would be 1/4 plus 1/4 would be 1/2, and that's where I get that ratio. Okay, so I'm gonna stop here, but I want you guys to if you have time in your busy schedule over the Thanksgiving break, to work out these problems, because when we come back, we'll discuss what the answers are. But you should be able to answer them pretty quickly, right? If you know the ratios. If you don't, you're gonna have to make Punnett squares. So what's the odds of getting a purple flower from the cross, big P, little p, big P, little p? What kind of cross is that? Monohybrid. So so I'm asking you, purple flower, what's purple flower? Phenotype or genotype? So what's the ratio? So what's the odds of getting a purple flower? 3/4, right? Or 75%. What are the odds of getting a big P, little p genotype? You know, the ratio of genotype is 1 to 2 to 1. So what's big P, little p? So it's 2, 1/4, 2/4, 1/4, right? Because there's 4 outcomes. So it's 2/4, which is 1/2. Excellent. What about little p, little p? 1/4, right? So either you know the ratios or you're going to be writing out Punnett squares. Now on the final, that might not matter because you're going to have 2 hours to do it. But you're not going to want to write out 16 square Punnett squares. It's not fun. And then what if you had to write out a 64 square Punnett square? That would take you the whole test period, probably. So what about this one? What are the odds of getting a wrinkled green seed from this cross? What kind of cross is this? Dihybrid. So wrinkled and green, what are those phenotypes? They're both recessive, so what's the odds of getting both recessives? Remember, it's 9 to 3 to 3 to 1. So add them up. What's 9 plus 3 plus 3 plus 1? So then it's 1/16. what are the odds of getting a round green seed phenotype? What's round? Dominant or recessive? And what's green? So then what part of that 9:3 to 3:1 ratio is 3/16, right? Or you could do the Punnett square, draw it all out, and then figure out the phenotypes from the genotypes. And by then your test may be over. So it's in your best interest to figure out. And then big R, little, big Y genotype. Yeah, so we could do the math, right? What's the probability if you do this that you get big R, big R? Remember, that's a monohybrid cross, so it's 1 to 2 to 1 genotype. So that's 1/4, and this one is 1/4. 1/4 times 1/4 is 1/16. So there you go. I just did it for you. You don't have to do it now. And we'll talk about the rest of this stuff later. All right. So how do you guys want to do this? So the test is chapter 12, 13, and 10, 12, and 13. So 10 is photosynthesis, 12 is mitosis in the cell cycle, and then 13 is meiosis and the formation of granules. Do you want to just ask questions like we did last time?
Yeah.
All right. So remember, we didn't cover that last section on cancer. We did talk about the 4 things that were required to cause cancer: tumorigenesis, metastasis, angiogenesis, and immortality. But you guys don't need to know any of that other stuff, like the stuff on the study guide. Like, we never talked a lot about P53 or treatments for cancer or any of that stuff. So you're not responsible for any of that.
Any of the cancer stuff.
Right, because in none of those slides we covered in lecture. I mean, except for a couple. So you need to know that cancer cells, right, most cells are anchorage dependent and density dependent, but cancer cells, they don't care. And that the 4 things that make cancer dangerous are creation of a tumor, so uncontrolled cell cycle, tumorigenesis, metastasis when it spreads around, angiogenesis by recruiting new blood vessels that can feed itself, and then immortality is the last one. So that's really all you need to know. And that's all on a slide, all on one slide, so you can look at it. All right, so organic carbon in a tree comes primarily from what source? It's not a trick question. We talked about this. So what do plants utilize as their main carbon source? I mean, we talked about it. Your Roundup that you spray on the plants kills them because they utilize this main carbon source for everything. To make proteins. What do they make the proteins out of? Do you remember? They have to make it out of the sugars. So the question is, how do they make sugars? What's the source of the carbon to make sugars? Because that's where plants make all of their stuff. No. I'll give you a hint. It's the dark reaction. That's where they make sugars. Light doesn't have carbon in it. Water doesn't have carbon in it. Water is H2O.
Air. CO2.
It's CO2, right? Remember, what do plants need for the dark reaction? I told you guys, you need to know what goes in and what comes out. So what do they need for the dark reaction?
Carbon dioxide.
3 CO2s, 9 ATP, 6 NADPH. Sure is a good thing we didn't have the test today. So this makes glyceraldehyde 3-phosphate in the Calvin cycle, which is also known as the dark reaction. So the source is carbon dioxide. What experiment would produce heavy oxygen as a gas? So remember we have different carbons, carbon 12 and 13 and 14. What do we call those? Isotopes. So we have oxygen. What's the regular mass of oxygen? This one's 18, so what does that mean? One neutron? 2 extra neutrons, right? Because a neutron weighs 1. So it's the same element, it just has a different mass. We can measure that because it's heavier. That's what I mean when I say heavy oxygen. So which one of these experiments would produce oxygen as a gas? If we had water that had the heavy oxygen in it, or if we had CO2 that had the heavy oxygen in it. And what you have to ask yourself is, where is the oxygen produced from the photosynthetic reaction?
Air.
No.
Oh my gosh.
It is the light reaction. Now we're narrowing it down. Where's the source of the oxygen? What did you say? Water? Why do you say that? All right. I can sense that we need a review on the light reaction. So what's the first photosystem? Photosystem 2. Remember, it has a antenna complex. What's in the middle of that antenna complex? What's the reaction center? It's what? It is, but what's the chlorophyll? What kind of chlorophyll is it? It's called P680. It's chlorophyll A. And why is it called P680? Because it works best at 680 nanometers. So there's external accessory pigments. What are those? No. Accessory pigments. We separated them out in the lab. Carotenoids and so part of the antenna complex that helps the reaction center is chlorophyll B and the carotenoids. They focus the light on A. What happens? Magnesium gets excited. The electrons jump. How many electrons in magnesium? 2. Excellent. Those electrons are missing. How do they get filled? That's right. Water, H2O gets split, the electrons get filled, and what gets released? Oxygen. So what's the correct answer for this then? Experiment 1, that's right, because it's in the water. CO2, if I asked you, well, we did this one, where would you expect the heavy oxygen to end up? Well, come on, we just went over it 2 seconds ago. Yeah, because the dark cycle requires what? CO2 and what comes out, which is made into glucose. So you would expect that would end up in G3P, right? In fact, this is how scientists discovered half of what's going on inside the photosystems in the dark in the light reaction. So where do these electrons go now? They go to photosystem. Excellent. And there's another antenna complex. Accessory pigments, what are they on the outside? Yes, B and carotenoids. Excellent. They're focusing the light on the reaction center A chlorophyll, which is called P700. Why is it called that? Works best at 700 nanometers. So the light's focusing on that. What happens to the magnesium? Excited. Remember chapter 2, electrons jump to a higher energy level. How many electrons?
2.
Excellent. And that ends up on NADP plus to make NADPH. Good. Where are these missing electrons come from to get filled in? No, they came from photosystem 2. Yeah. So this thing sends electrons here, there, and what kind of electron flow do we say if it's direct? It's non-cyclic. And remember, how many, if we have 2 electrons, how many protons get pumped across that integral membrane protein? 2 protons. How many protons do we need to make an ATP? 2. So how many ATPs do we make from this? 1 ATP. Excellent. And if it goes on to here, how many NADPHs do we make? 1. So that's a 1-to-1 ratio, right? But remember I told you that we need 9 ATP to 6 NADPH. Is that a 1-to-1 ratio? What do we need more of? ATP. So we have to send the electrons back through the first electron transport chain. And when we do that, what do we call that kind of electron flow? Yes. All right. So I have a crystal ball, and I sense some studying in your future if you want to do well on this test next week. Reaction center chlorophyll of photosystem 1 is known as P700 because correct answer is B, right? It's best at absorbing light with a wavelength of 700 nanometers. In a plant cell, where are the ATP synthase complexes located? So then the answer is D, correct. They're found in the thylakoid, right? In the membrane. And those are used for what reaction? Well, remember we have to feed that into the dark reaction to make the sugars. And then it's also in the mitochondria, and what are those ATPs for? The energy of the cell. We just covered that in chapter 9. Not in the plasma membrane. What organism does have it in the plasma membrane? Bacteria, that's right. All right, so where do the electrons that are found to replace those lost in photosystem 2 come from? Water. So it's D. Where's the Calvin cycle located? No. It's in the stroma. So the correct answer is B. Where do the so this is a typo. This should be photosystem 1. Where do the electrons to replace the lost electrons in photosystem 1 come from? No. No. It's photosystem 2, right? They come directly from photosystem 2.
So where does the Calvin cycle happen? In the
Calvin cycle is in the stroma, in the inner part of the chloroplast. What are the stacks of thylakoids called? Oh, sorry. It's blinking. I can't turn it on. Grana A, that is correct. Okay. So to do well in the test, you guys should be able to answer all of those questions. Nope. Okay, so this is chapter 12 questions. Which of the following best describes the kinetochore? All right, you got to decide. Time's running out on your test. Answer's A. Remember the kinetochores, the proteins associated on the outside near the centromeres, and that's the site of attachment for the microtubules.
How come B is wrong?
Because the centromere region of a metaphase chromosome in which the DNA combine with spindle proteins, it's not the
Because it's anaphase?
well, it's not just of the metaphase chromosome, right? Because it attaches in prophase, and it also is still attached in anaphase, so it's not exclusive to metaphase. All right, so during anaphase, what happens? Remember, this is mitosis. So that's correct. Remember, the chromosomes separate, the centromeres come apart. The first phase of mitosis is called what? Prophase. What happens in prophase? It comes apart. Centrosomes duplicate, move apart. What else happens? Chromosomes condense. That's in meiosis. So you're a chapter ahead. Yeah, because that's in your, so we covered that in mitosis. So cell division is completed when which step is finished? No, because after mitosis is cytokinesis, and that's when the two cells completely separate. So the correct answer is A. The point where the two chromatids are attached, what's that called? Right, that's the centromere, correct. During which phase does the nuclear membrane become degraded and microtubules invade the nuclear space?
Metaphase.
Prophase.
I think it's
C.
A, B, C, D, E.
All right, just guess.
I'll say A.
The correct answer is A. Prophase, the nuclear envelope is still intact. So it's not till prometaphase that the nuclear membrane comes apart. So make sure you guys go back and study that stuff. What's the order of phases during mitosis?
Is C. C. C.
The correct answer is
C.
C. Good. Chromatin is
C. C.
correct answer is C. Right? What are the proteins called? Histones, that's right. Which step is or are not a part of M phase?
C.
Yes, C, S phase, what is that part of? Interphase, good. Which checkpoint ensures that the spindle fibers are attached to the kinetochores? So remember there's 3 checkpoints, G1, G2, and M. Correct answer is M, right? Kinetochores are the points where the microtubules attach. So they line all the chromosomes in the middle. Remember they have to make sure they're all attached before they start to anaphase. So that's what that checkpoint's for. Which checkpoint makes sure the DNA is replicated properly? G2, yeah. What happens if they're not? Yeah, what do we call that? Apoptosis, good. Cellular suicide. And if they're okay, they meet the criteria, then they go through M phase. Excellent. What complex promotes that? What cyclin-dependent kinase promotes that? So remember there's a cyclin and a cyclin-dependent kinase that promotes that next phase, the M phase, and what is that called? All right, well, here's the question. So what cyclin-dependent kinase complex allows passage through the M checkpoint? This one's different, because I asked you the G2 one. What is it? Anaphase, so what's the letter? A? You guys looking at the same screen? But yeah, it's B. It's the anaphase promoting complex. Because the M phase arrest is at metaphase. What's next after metaphase? Anaphase. Now, the other question was what cyclin-dependent kinase allows passage through the G2 checkpoint? Yeah, that's A. And what does A stand for? Mitosis promoting factor, correct. All right. What transmits your genes from one generation of a family to the next? Yeah, it's gametes. Yeah, that's what passes on your genes. So, privet shrubs and humans have the same diploid number, 46. So what makes us different? What makes you different than a shrubbery? Yeah, you got different genes. That's exactly right. So the answer is D. During meiosis, the resulting gametes have blank the number of chromosomes as the parent cell. Yeah, correct. The answer is half, right? So you're 2 in, your gametes are in. How and at what stage do chromosomes undergo independent assortment? It is not C. It is not B. It is not A. Sure. So remember, independent assortment means that how they assort on the metaphase plate. And we went through that. That increases diversity. So remember we said that you could have
So the meiosis one metaphase.
you could have them line up like that, and those separate, and those would produce gametes that would look like this. One little one and one big one. Or this could flip, right? They could assort independently during metaphase 1, right, of meiosis. So this could line up like this, where the black ones are on this side, and the red ones are on this side. Sorry, the marker is lame. But you guys get it. So because of that flip, which is called independent assortment, these gametes are now look like that, which are different. So if you don't know what independent assortment means, then you wouldn't be able to answer that. And we covered that. That's one of the things that leads to variation. Remember we said there's how many tetrads in humans? So when we're before metaphase, how many chromosomes do we have? No. No. For metaphase 1, how many do we have? You have 46. You doubled your DNA in S phase. You haven't halved it yet. So what do you have? 92. So if you have tetrads, groups of 4, what's 92 divided by 4? 23. So you have 23 possible combinations, right? Which is 2 to the 23rd power. Remember when I told you that's roughly 8 million variations from independent assortment. Crossing over occurs during which phase of meiosis? Correct answer is A, prophase 1. Excellent. Which phase of meiosis is this in? How do you know it's anaphase 1?
Because there are
What if it was anaphase too? Well, yeah, there would be 2 sets or 4 sides, right? So yeah, so it's anaphase 1, that's great. Humans have 46 chromosomes in their somatic cells. This is an example of what number? Diploid, right? So remember our 2n equals 46, so n equals 23. Excellent. In this cell, what phase is represented? Correct answer C. How do we know? Well, it's not that they're crossed, it's because they're lined up as tetrads, right? There's 4 of them. If the answer was E, that wouldn't look like that at all, right? They would be all lined up as sister chromatids individually. There wouldn't be 4, there would just be 2s. So that's how you know. What phase is represented in this cell? It's not anaphase. Correct answer is E. Why?
So it's already been through.
Right, so we know it's meiosis 2 because it's already got through 1 division, and we know it's metaphase because it's lined up in the middle. And you guys doing that, if you haven't done it already, you looking at those slides are going to help you decide what phases it in. What about this one? Correct answer is D. Why? There's 4 of them, and you can see the cleavage furrows, right? You guys see those? Right here, and here, and there. And that's a sign of telophase. So Taxol, this is true, is an anti-cancer drug extracted from the Pacific yew tree. In animal cells, how Taxol works is it doesn't allow the microtubules to disassemble, so the tubulin, it would be like gluing all the Lego blocks together. So why does that cause them not to be able to move during metaphase, and they can't go into anaphase? Remember what happens is the chromosomes walk, and then the tubulin comes apart at the end. So if they're on a rod that can't disassemble, what happens? They get stuck and they can't move. So then it arrests, and that causes the cells to terminate in that phase. And so they eventually die because they can't read their genes or anything. So the answer is A, right? Because what are the fibers of the mitotic spindle made out of? Tubulin. Excellent. In which group of eukaryotic organisms does the nuclear envelope remain intact during mitosis? So just to remind you, the answer is B and C. We talked about that. So it would be B. All right. Any other questions? Make sure you guys study. You got 5 days before the test. Just shove it all in there. I'll see you on Tuesday. Happy Thanksgiving. Try not to be so depressed. Yeah, and just to remind the hybrid class, your test is on a Wednesday.